Joe and I met in 1966 when we began our internships at the Massachusetts General Hospital in internal medicine. I had been a medical student at the University of Pennsylvania, the nation's oldest medical school, and Joe had graduated from Southwestern Medical College in Dallas, Texas, the nation's youngest medical school that I had never heard of. I thought it was a Bible school. But within 2 days after we began our internship, we realized that we had something in common. We were both interested in the fundamental basis of disease, not only in taking care of our patients, which was our first responsibility, but also trying to figure out why this person had a heart attack, why this person had rheumatoid arthritis, why did this person have pneumonia. And it was that interest in the basis of disease that created this friendship that has now lasted since 1966. After our residencies, we went to the National Institutes of Health, where both of us worked with very exceptional scientists, Joe with Marshall Nierenberg and I with Earl Statman, two of the best, most brilliant scientists at NIH. But we also had clinical duties. And there we saw a patient that actually determined the scientific course of the rest of our lives.
So the patient that Mike and I saw at the NIH was a little girl, the first patient, with a disease called familial hypercholesterolemia, also called homozygous familial hypercholesterolemia, or FH. And her brother actually had the same disorder. And this little girl has a very high cholesterol. Her cholesterol is as high as 800 to 900 milligrams per deciliter. The normal of a child of this age would be about 120 milligrams per deciliter. And most of this high cholesterol was in a lipoprotein called LDL, which is known to everyone as the bad form of cholesterol. And when LDL accumulates in the blood, as in this little girl, it deposits in the skin in these little yellow lumps you see on the surface of her skin that cause xanthomas. And it also accumulates in the artery wall and produces coronary heart disease. And this little girl had a heart attack when she was 6 years of age. Now, this little girl had only elevated LDL. She had no high blood pressure. She had no diabetes. She didn't smoke. She didn't have a type A personality. Her only risk factor for having a heart attack at age 6 was this high level of LDL. So this is one of the best examples of a disease where we really know the cause, and the cause of the disease in this little girl is a increased level of her LDL. And so it will be important to understand the basis of why her LDL is elevated and also how it causes her disease.
This is LDL, the lipoprotein that builds up in the plasma of this young girl. It's a spherical particle that's about the size of a small virus, and each LDL particle has 1,500 molecules of cholesterol within its core, and it's surrounded by a polar coat of phospholipid and a protein called ApoB. And LDL circulates in the blood on average for about 3 days in humans, and eventually it leaves the bloodstream. When we began our work, no one knew anything about what controlled the level of LDL in the blood. We were interested in this disease because not only do the homozygotes have problems, but the heterozygotes, people who inherit one copy of the abnormal gene, have a moderately elevated cholesterol, and they have heart attacks not at age 6, but at age 60. And so if you ignore the homozygotes, the heterozygotes transmit their disease in an autosomal dominant fashion through the pedigree. And so having a single copy of the mutant gene, no matter what it is, produced a disease. Now at the time, there were about 35 single gene diseases that had been worked out, and the defective gene product was known. All 35 of those were enzymes, mutations in enzymes, and all of them were autosomal recessive. That means that the heterozygote had no symptoms. But in the case of familial hypercholesterolemia, the heterozygote had a disease. And we thought that if we could understand this disease, it would be the first dominant disease that could be elucidated in humans, and it might teach us something about proteins that are not enzymes. We assumed that it was not going to be an enzyme, and that it would probably be some disorder of regulation. Now, Joe had promised his professor at Southwestern Medical College that he would return there after his training. And Joe and I talked about this, and we decided that I would also go to Dallas. And when we went to Dallas, we would both try to figure out what type of defect could cause this high LDL level. And the unique approach that we took was to study the metabolism of cholesterol in cultured cells. We obtained biopsies from children with familial hypercholesterolemia and compared them with normal cells and studied the regulation of cholesterol synthesis because we thought that this would be some regulatory abnormality.
So here's an experiment that Mike and I carried out in 1973, and it's arguably probably the most exciting experiment that we've ever done, and it really hooked us on science and also taught us a lesson that I'll come back to after describing the experiment. And so as Mike pointed out, we wanted to understand what there was about the regulation in cholesterol synthesis in these children with homozygous FH that was different from normal. And so at the time, it was known from studies in rats and mice that when you fed one of these small animals cholesterol, you would suppress cholesterol synthesis in the liver. There was a hint that there was a regulatory system. Well, and that the liver was known to be the most important organ in the body for controlling cholesterol, but there was no way that we could get liver biopsies from patients. So we chose fibroblast, which one could get, skin fibroblast, and this was sort of a heretical idea at the time that one would study a fundamental process that was going on in the liver in a fibroblast. But we did it anyway, and here's the experiment. And the first experiment we did worked, and it was really quite dramatic. So what we did is to compare fibroblasts grown in fetal calf serum, which contains LDL from a normal subject and one of these children with homozygous familial hypercholesterolemia. And you can see in the red curve on the left that the level of the cholesterol synthesis was very low, almost undetectable. Now, as a surrogate for cholesterol synthesis, we used the enzyme HMG-CoA reductase, which had been known from these animal studies to be a key rate-controlling enzyme in the cholesterol biosynthetic pathway. So we were able to set up an assay for this enzyme, and we would, after incubating the cells with this fetal calf serum that contained LDL, we would make extracts and do the measurement. So you can see that at 0 time, in the red curve, the level was very low. And then when we removed the serum and the LDL from the media over the next 24 hours, the enzyme activity in the normal cells in red went up 50 to 100-fold. And then when we, on the right panel, when we added LDL back, you can see that very high level of the enzyme HMG-CoA reductase came down to the basal level when the LDL was added back. So this in itself was a very interesting experiment in the normal because it told us that there was an effect of LDL cholesterol on regulating the activity of this enzyme that reflected the cholesterol biosynthetic pathway in normals. But that in itself was exciting, but the real dramatic result was when this result in the normal was compared to the FH homozygote shown in yellow. And you can see that at 0 time, in the presence of fetal calf serum and LDL, the level of the enzyme HMG-CoA reductase and cholesterol synthesis was 100 times higher than in the normal. And it didn't change no matter what we did. When one removed the LDL, it stayed high, and when added LDL back, it stayed high. So this was really dramatic. It was really all or none, and that was one of the things that I think not only was it an exciting result, but it sort of addicted us to the fact that we were really interested in all future experiments and seeing big changes. We don't usually see changes of 100-fold, but at least it told us that one had to have really a big change to really work out something. Now, our simplest interpretation from this result, when Mike and I sat down and tried to deal with it after 24 hours of being all on the wall and off the wall of excitement, we decided that the simplest interpretation was that the FH homozygote cells had a mutation in their enzyme HMG-CoA reductase that rendered the enzyme resistant to feedback regulation by cholesterol. So we said, okay, well, if that's true, then we should be able to add cholesterol not in LDL but in ethanol, and we would get the same result. But then when we did that, the curve, the yellow curve in the FH homozygote came down from this 100-fold, just like the normal. And so the normal and the FH homozygote cells are now responding to cholesterol and ethanol, whereas the FH homozygote did not respond to the LDL. So that then told us that our original hypothesis was wrong, and now we have a new hypothesis that said that the defect in the FH homozygote was a failure to extract the cholesterol from LDL. So how does a normal cell, this now became the question, how does a normal cell extract the cholesterol from LDL?
So we radio-labeled LDL with iodine-125 and incubated it with fibroblasts. And what we saw was that when we incubated the LDL with normal fibroblasts, there was high affinity cell surface binding that was saturable, as shown by the red line on this slide. But when we incubated the LDL with the FH homozygote fibroblasts, there was no binding. So the problem was that there must have been a receptor on the cell surface that binds LDL, and the FH homozygote cells lacked this receptor. Now, this was 1974. It was in the very, very early days. Receptors were kind of theoretical concepts. How they worked was, there was very little known about how they actually worked. But we went one step further when we incubated the cells with I-125 LDL. We followed what happened to the LDL after the binding reaction. And we were shocked to find that the I-125 LDL was totally degraded. The protein was degraded down to amino acids. And the only way that could happen would be if the LDL had been taken into the cell and degraded in the digestive organelle of the cell, which is the lysosome.
So once we knew that there was a putative receptor on the surface of the cells, we advanced the concept of an LDL receptor pathway in which LDL bound to receptors on the cell surface in specialized regions called coated pits. And our colleague and collaborator Richard Anderson was able to demonstrate by electron microscopy the localization of ferritin-labeled LDL in these coated pits. Coated pits occupy only about 2% of the cell surface of a fibroblast, and so it's a very specialized region for rapid endocytosis and internalization of proteins like LDL. So once the LDL bound to coated pit, the coated pits pinched off to form coated vesicles, which delivered the LDL to endosomes and then ultimately the lysosomes, where the LDL was degraded and the cholesterol was released, and that cholesterol was then used by the cell for membrane synthesis. So we now knew from the LDL receptor pathway that in addition to obtaining cholesterol from cholesterol synthesis, cells could also obtain cholesterol through the receptor-mediated endocytosis of LDL. And this cholesterol and not only was used for the synthesis of plasma membranes, but also it exerted a feedback regulation on the enzymes involved in the cholesterol biosynthetic pathway, and it also exerted a feedback suppression on the gene for the LDL receptor. So one had a coordinate feedback regulatory system based on both entry from LDL and biosynthesis. So what happened in the FH homozygote cells that lacked LDL receptor? How did they obtain their cholesterol for membrane synthesis and maintain cholesterol homeostasis? So in these cells, they rely virtually completely on the cholesterol biosynthetic pathway, and that is their source of cholesterol. So the cell is happy because the cell has its cholesterol for its membrane synthesis, but it's all at the expense of an elevated LDL in the bloodstream, which is the root of this disease, familial hypercholesterolemia, when the LDL builds up and then ultimately deposits in the skin for these xanthomas and in the artery wall to produce atherosclerosis.
So over the next few years, we were able to define in detail the pathway by which LDL delivers the cholesterol to cells, and it's shown on this slide. After binding to the receptor-encoded pits, the LDL is internalized and it's delivered to endosomes. And within endosomes, there are proton pumps that acidify the interior of the endosome. That causes a conformational change in the receptor so that it releases the LDL particle. The LDL is then carried further into lysosomes where it's totally degraded and the cholesterol is liberated for use to make new membranes. LDL-derived cholesterol is also used for steroid hormones, bile acids. It's used to make new lipoproteins, and as Joe mentioned, it exerts regulatory action. So the LDL-derived cholesterol is really important biologically. After the LDL dissociates from the receptor returns to the cell surface in a recycling endosome. And each of these, each LDL receptor makes one trip into and out of the cell every 20 minutes. And it makes hundreds of trips during its lifetime. And each time it carries an LDL particle with 1,500 molecules of cholesterol ester. We gave this process its general name, which is receptor-mediated endocytosis. And after we had described the system for LDL, it turned out that other scientists found that other proteins bound to their own receptors on the surface, but they were taken in and into the cell in the same way as LDL. And the examples include hormones such as insulin, transport proteins such as transferrin, which transports iron and vitamin, and the protein that transports vitamin B12. So receptor-mediated endocytosis turned out to be a general property of all cells, at least all nucleated cells. And it's interesting that we had no idea that we would stumble into something that was so general, because all we wanted to do was figure out what was wrong with these children with FH. So, you know, this slide shows what we looked like when we started and what we look like now. I think it's important for any young people looking at this video not to picture us the way we are now as shown on the right, but picture us as we were in 1972 when I was 31 years old and Joe was 32 years old. That's when we made these big discoveries, and the thrill of discovery when you're young and working hard and something happens like that FH experiment that Joe described, the thrill of that is amazing. And I want to say one other thing, that it's a fantastic thing to share that experience with somebody else. We have the longest running partnership in we think in the history of science, to be honest. And I just have a hard time understanding why somebody would want to do science totally on their own without somebody to share the adventure. You wouldn't want to take a trip to Paris by yourself, would you? I mean, you want somebody along who can share the excitement of that adventure, and that's what Joe and I have had. So it's been it's a fantastic thing to have a partnership in science.